will answer the question number 1. I found in the answer in the book Moran Horton. So the book answer is some species cannot synthesize the carbon skeleton of every amino acid. Mammals, for example, can make only about half of the amino acid they require. The rest, called essential amino acid, must be obtained from the diet. Non-essential amino acids are those that mammals can synthesize in sufficient quantity, provided they receive adequate total dietary protein. So from the sentence state above, I can conclude that essential amino acids are amino acids that must be taken from food or other intake depending on amino acid that cannot be produced by the body, or only amino acid that can be obtained, while non-essential amino acids are acids that can be synthesized by mammals in sufficient quantity, provided they receive adequate total food protein. So in here our body need essential amino acid, but the body cannot to produce that. So we can get the essential amino acid from the what we eat, apa yang kita makan. So this is which include essential amino acid: valin, histidin, tryptofan, isoleusin. So valin example spinach. We can get the valin if we eat spinach or ingredients anymore and other. And then histidin, example chicken. And you can see in my slide, this slide. And then this is include non-essential amino acid: proline, serine, alanin, arginin, asparagin, aspartic acid, and other.
So, the question for number 2 is, please explain the amino acid metabolism and the nitrogen cycle. So, there are 2 arrow paths, this one and this one, that can convert the nitrogen into ammonia, which are directly and indirectly. As we know that nitrogen is chemically unreactive because it has a strength triple bond. So, if we want to convert this into ammonia, there are some steps that has to be gone through first. The first one is the direct arrow path. This path only can be done by bacteria like cyanobacteria and soil bacteria. So this bacteria has an enzyme called as this nitrogenase. So in here, the direct path can be only done if there is a nitrogenase enzyme. The nitrogen converts directly into ammonia with the help of nitrogenase enzyme. This process called as the nitrogen fixation. And after that, for the second pathway is this pathway, the indirect pathway. Why indirect? Because there are several steps that the nitrogen has to pass through first, so it can become ammonia. So here, the nitrogen is oxidized into nitrate and nitrite, which is catalyzed by the high voltage during lightning storms. The nitrate that produce this, the nitrate that is produced, then converted into the nitrate. The nitrate converted into the nitrate with the help of nitrate reductase. And the nitrate is helped by the nitrate reductase to form this ammonia. Either from the direct or the indirect aerob path, the ammonia that is produced, then will we go to the metabolism pathways, and after that it might produce the amino acids, nucleotides, and also phospholipids.
In the nitrogen cycle, there is some term. The first term is fixation. Nitrogen fixation is reduction of nitrogen to be ammonia in a process. Ammonia from decayed organism is oxidized by soil bacteria to nitrate. The formation of nitrate we call is nitrification. Some anaerobic bacteria can reduce nitrate or nitrate to be nitrogen. This process we call is the nitrification.
Okay, I will explain about assimilation of ammonia. Ammonia is assimilated into a large number of low molecular weight metabolites, often via the amino acids glutamate and glutamine. The reductive amination of alpha-ketoglutarate to glutamate by glutamate dehydrogenase is one highly efficient route for the incorporation of ammonia into the central pathways of amino acid metabolism. The glutamate dehydrogenase of some species or diseases are specific for NADH, while others are specific for NADPH. If alpha-ketoglutarate react with NH4+ with NADPH and plus H+, will be produced NADP+ plus H2O, and the product is glutamate. If the glutamate react with glutamate dehydrogenase, will be produced alpha-ketoglutarate. Between alpha-ketoglutarate and glutamate occurs the reversible reaction. The glutamate, if we react with NH4+ and ATP with glutamine synthetase enzyme, will be produced ADP and pyruvate, and the product is glutamine. In mammals, glutamine carries nitrogen and carbon between tissues in order to avoid high level of toxic NH4+ in the bloodstream. The amide nitrogen of glutamine can be transferred to alpha-ketoglutarate to produce two molecules of glutamine in a reductive amination reaction catalyzed by glutamate synthetase. This is the reaction. Glutamine react with alpha-ketoglutarate with glutamate synthetase and help of NADPH and H+ will be produced NADP+, and the product is two molecules of glutamate.
Assimilation of ammonia into amino acid. The first, glutamate dehydrogenase pathway. Alpha-ketoglutarate. To yield glutamate that can undergo oxidative deamination, catalyzed by glutamate dehydrogenase. And then, the transmination occur in the opposite direction, when the amino acids are being active formed and the amino group are donated by glutamate. Next, glutamin synthesized and glutamate synthesized under condition of low ammonia concentration. In bacteria, the glutamate dehydrogenase use the couple reaction catalyzed by glutamin synthesized and glutamate synthesized for the assimilation of ammonia into glutamate. the conversion of alpha-ketoglutarate to be glutamat by the glutamat synthase, and the glutamin to be glutamat by the glutamin synthase. After this formation, the glutamat undergoes transmination with the alpha-keto acid to form amino acid.
Answer the number 4. I found this answer in the book transaminase. You can read this, but I will read. The amino group of glutamate can be transferred to many keto acid in reaction catalyzed by enzyme known as transaminase or aminotransferase. The amino group of glutamate is transferred to various keto acid, generating the corresponding amino acid during amino acid synthesis. Most of the common amino acid can be formed by transamination in amino acid catabolism. Amino groups are transferred from various amino acids to a ketoglutarate or oxaloacetate, generating glutamat or aspartate. So, from this, I can conclude that transaminase is an enzyme that transfers the glutamic amino group into the form of a keto acid. In amino acid catabolism, amino groups are transferred from various amino acids to a ketoglutarate or oxaloacetate, which produce glutamate or aspartate. So, transaminase can change amino group of glutamate to be many keto acid. And then oxidative deamination. Oxidative deamination is the release of amines from glutamate which produce amino ions. And this answer I found in PDF Biokimia Biologi Unut. Okay, you can see in the picture in the left, delta amino acid. If you delta with transaminase reaction, alpha amino acid can transfer into delta keto acid, and the delta keto acid can change form into alpha amino acid too. And the picture right, you can see transamination and oxidative deamination. So transamination example is alpha ketoglutarate from delta ketoglutarate become glutamat, and oxaloacetate become aspartat. Nah, aspartat become to the area cycle. And oxidative deamination, you can this picture right and the right, NAD plus H2O or water become NADH plus NH4 plus